Hello all and welcome to our presentation on genetic engineering. So on this slide over here, you see a question. Is genetic engineering or genetically engineered food frankenfood or is it a hunger stopper? And when I say frankenfood, we mean is it some kind of scientific, spooky, scary food that we genetically engineer our food? Or is it something really useful that can probably be used to solve worldwide hunger? So let's look into this. How do we move the slide? So genetic engineering is a technology that allows humans to alter the genetic makeup of organisms. That means we can change the genes of an organism. So back in the day, and we talked about this with Mendel and with reproduction, farmers were always able to take two different species and mate them together. Like an apricot is an apricot a peach and a nectarine together? I don't think so. But there's a lot of foods that are made by crossbreeding. Sometimes a farmer would take his best growing orange and only plant those orange seeds so that he would always have good fruits. We might cross in nature. Sometimes we cross animals, right? We spoke about crossing a horse and a donkey to produce a mule. And then we have, desirable traits in the mule. But that's different from genetic engineering. In all the examples I mentioned before, we're mating either organisms from the same species or very closely related species so that they have certain traits in their offspring. When we talk about genetic engineering, though, we're simply taking a gene from one organism and sticking it into a gene from another organism. So you could theoretically take a gene from a fish and stick it into a tomato plant, and then you'd have a certain trait in the tomato plant. So this allows us to do a lot more than just crossbreeding. So for example, we could have designer plants. So here you have a plant that, as you can see, is glowing in the dark because scientists inserted the gene for luminescence or for glowing from a firefly into the genes of a plant. In this case, here we go. You have a gene that makes stronger wood. That's practical. And in this case, you'll notice you have a green plant and then a nice dried, shriveled up, nasty brown plant. And that is because there's an insertion of a pesticide-resistant gene. So this last example is really common and very beneficial to us, where if you take it away and you look, in this green plant, we put a gene that makes this plant resistant to pests. Certain bugs will not eat this plant now because it has this gene, whereas this plant does not have that gene, and you could see this plant is much more beautiful and healthy. Here we go. In recent years, new varieties of farm plants and animals have been engineered by manipulating their genetic instructions to produce new characteristics. So what are some of the characteristics that we would want in plants? We want our plants to survive the winter. We want our plants to not be killed by disease. We want our plants to not be eaten by bugs. And if we genetically engineer a plant and we put a gene for disease resistance into a plant, then we don't need to spray the plant with chemicals because it's already resistant to the disease. So in the United States, almost all of our soybeans are genetically modified. A lot of our wheat and corn is also. So how does it work? How are we able to insert genes from one organism into another organism? So I'm going to put everything up and we'll go through it here. So let's look at our picture here. And in this example, we're talking about genetically engineering a bacteria so that it can produce human growth hormone. And what happens here is basically what happens in all types of genetic engineering. So the first thing we do is we isolate the gene that we would like to copy. So in this case, we want the gene for human growth hormone. So what you could see is that the gene for human growth hormone was cut out of the DNA of a human cell. So now I have just here my gene from human growth hormone. And then I have another cell, a bacteria cell, and my bacteria has a plasmid, a round piece of DNA. And I'm going to combine my human growth hormone gene with my bacterial DNA. and I'll be using special enzymes called DNA ligases to glue the human growth hormone into the bacterial DNA. And my combined growth hormone and bacterial DNA is called recombinant DNA, right? It's a combination of human growth hormone and a bacterial plasmid. And then I'll use something called a vector to stick this recombinant DNA back into a bacteria. Now, based on what we know about genes, as long as an organism has a gene for a trait or for a protein, it should be able to make that protein. And that's exactly what happens. We take this recombinant DNA, stick it into a bacteria, and now our bacteria can produce human growth hormone. So the bacteria isn't human, but it's able to produce human growth hormone. And over here we have a freaky example again, similar to the first one we saw, where this monkey is now able to glow in the dark, basically, because it has the gene from a firefly that allows the firefly to glow in the dark. But going back one second here, just to our recombinant DNA. So if we were to talk about a pesticide-resistant plant, so there are some bacteria that are resistant to diseases that harm plants. So we would take that gene for disease resistance from the bacteria, combine it with the plant's DNA, and then that plant would also start to be resistant to disease. And this is a technology that's really, really becoming more popular and really useful. One of the most amazing uses of genetic engineering is that we're able to produce bacteria that produce human insulin. And we all know that diabetes is a major problem. So following this same procedure, we're able to cut out the gene for human insulin, combine it with the bacterial DNA, stick it in bacteria, and now the bacteria produces human insulin, which is amazing for the large population of Americans who suffer from diabetes.